We shall begin our examination of a theoretical proposal that connects the biological structure of the genetic code to the abstract domain of hyperbolic geometry as conceptualized within the TGD framework.
And this brings us to a rather profound question. Is the code of life merely a product of stochastic chemical events, or could it be an expression of a much deeper geometric order?
Here is our path for this exposition. We will begin with the geometric basis for the idea, then examine the specific icosa-hedral tetrahedral tessellation. From there, we'll see how this model maps geometry to codons, explore its predictive power regarding symmetry and degeneracy, and finally, consider its proposed physical realization and the implications for a new quantum biology. So, let's delve into the core premise. This model posits that the structure of the genetic code is not arbitrary, but is in fact a direct consequence of a specific geometric tessellation. It's quite important to note this is a model inspired by the TGD framework. TGD itself does not fundamentally rely on this specific tessellation. Here is the central hypothesis, stated formally. The 64 codons of the genetic code are posited to be realized by the structure of an icosa-hedral tetrahedral tessellation, or ITT. And this structure doesn't exist in our familiar Euclidean space. It resides in what's known as hyperbolic 3 space, or H3. Let us now transition to the geometric foundation of the model. We're looking at a unique tiling of hyperbolic space, a kind of honeycomb composed of Platonic solids. It's crucial to understand the primary components. The ITT is constructed from 3 types of Platonic solids: the 4-faced tetrahedron, the 20-faced icosahedron, and the 8-faced octahedron. In this particular model, the octahedra are considered passive, almost like voids or spacers within the structure. The active information-carrying components are the tetrahedra and the icosahedra. To see how this geometry relates to the genetic code, we must consider the core structural unit of the model. At its very heart, it is anchored by a central tetrahedron. Now, this is the key geometric relationship. 3 icosahedra are arranged around that central tetrahedra, and each one of these icosahedra shares one of its faces with the tetrahedron. This arrangement, which we'll call a T+3I unit, forms the geometric basis for the code. As this quote from the source material underscores, it is this specific T+3I arrangement that is proposed to provide the direct structural realization of the genetic code. Now we arrive at the central claim of the model, how this precise geometric structure maps, number for number, to the 64 codons. The derivation is quite direct. We begin by accounting for the faces of the 3 icosahedra. Each icosahedron has 20 triangular faces, so 3 of them contribute a total of 60 faces to our count. Next, we simply add the 4 faces of the central tetrahedron. This brings us to the final tally. The summation is remarkably straightforward. 60 faces from the 3 icosahedra plus the 4 faces from the central tetrahedron yields a total of 64. This one-to-one correspondence between the geometric faces and the genetic codons is the numerical cornerstone of the entire model. So the number matches. But the model attempts to go further, to explain the phenomenon of codon degeneracy. To do this, it introduces a dynamic element: Hamiltonian cycles. This is what provides the model with its principal predictive capacity. So what is a Hamiltonian cycle? Well, you can think of it as a path traced along the edges of one of these polyhedra. The path must visit every single vertex exactly once before returning to its starting point, forming a closed loop. These cycles are governed by specific mathematical symmetries, characterized by discrete groups like Z/N. And here we have what the author terms the real news. The model predicts that if one identifies the orbits of the triangular faces, that is the codons, under the symmetries of these Hamiltonian cycles, one can reproduce the known codon degeneracy for amino acids with surprising accuracy. This is the model's key prediction, moving it beyond a simple numerical coincidence. This leads to a rather elegant conceptual link with music theory. Each codon, represented by a triangular face with its 3 vertices, is analogized to a musical 3-chord. A complete Hamiltonian cycle, which connects all the vertices, therefore defines an ordered sequence of these chords, a sequence the model terms a bio-harmony. 3 such icosa-tetrahedral harmonies, plus a smaller tetrahedral one, are proposed to account for the entire set of 64 codons. Thus far, our discussion has been confined to abstract geometry. We must now address the critical question, where, within a biological system, could such a geometric object be physically instantiated? And this truly is the pivotal question. An elegant mathematical model is one thing, but for it to be relevant to biology, it must have a plausible physical substrate. To answer this, the proposal invokes several core TGD concepts. It suggests this geometric structure doesn't exist within the physical DNA molecule. Instead, it's posited to exist at a magnetic body, a field body associated with the DNA. This field body is thought to be composed of monopole flux tubes carrying what are called dark protons. In this view, triplets of these dark protons at the vertices of the geometric faces represent dark codons, which then orchestrate the behavior of ordinary DNA. Finally, let us synthesize these concepts and briefly consider the broader implications of this TGD-inspired paradigm for quantum biology.
So to summarize the logical progression of the model, it begins with a geometric foundation, establishes a direct numerical correspondence to the 64 codons, employs dynamic symmetry to explain codon degeneracy, and finally proposes a physical substrate at the magnetic body of DNA. We conclude not with a statement of fact, but with a provocative question for further contemplation. If life's fundamental code is indeed underpinned by a universal geometry, what other biological phenomena might also be governed by its principles?